So good evening everybody, this is Tarun Tiwari. Yeah, so video is running, so we'll start the session. So yeah, today we are here just to discuss questions. So as you can see in the thumbnail, we are going to discuss the questions. I will just go in brief what is this question about and why we are going to discuss now. So in learning life, I started learning life very long back, like 2019, but due to so many reasons I couldn't continue that. But now I have reframed everything and I started again with this huge energy and enthusiasm, so that I can succeed in this field and I can serve students better. So in that part I have started one NEET crash course and the test series, and the first week is over. So I started the test series started in December 4, like Wednesday, and this is like Sunday. So Thursday, Friday, Saturday, after third 3 days. So today is the fourth or fifth day, that is the discussion day what we have. So here, as I promised you, we will discuss the questions of botany and zoology, which you wrote on Wednesday. So many students are still writing. Because of your request, I extended the time duration up to 6:30, and now I think time is over. So we will discuss the questions. Let me analyze what is the performance, and then we will start discussing the questions. So it will take 2 more minutes. So in that time period, I would like to talk something very important, and that will be your what is learning life and what we do in learning life. Learning life, I started like we can say 2019. I posted my first video 2019, and way back I posted one more video, but that's not very, we can say, that effective. So I posted my first video, I think general talk video, on one day before the NEET exam in 2019. 2019 2019, one day before the NEET exam. So after that I posted one more video and so on. And 2020 lockdown is I completed so many different sessions of different institutes, and in 2020 I started creating videos rigorously, and in that we have our own website also, www.learninglifeonline.com. We have our website, and we have our Telegram channel also. But this website, because of long gap, this website is not in use right now. But yes, within a span of time, like within few days, we will start our website. We will, renew our website at a link, that is www.learninglifeonline.com. If you once we start again, that in that website we have so many videos, and we have two channels in that website. One is foundation biology and another one is medical lectures. This medical lectures is for all NEET aspirants, that is those who are in 11th standard, those who are in 12th standard, and all first year MBBS students, those who are like preparing, I mean those who are in their very first year. So all the fundamentals, all the basics of medical physiology, molecular biology and cell biology, biochemistry, all these things will be there in this video channels. So these two channels we have, and one is foundation biology and another one is medical lectures. This is available in our website, and we will, like as I told you, again we will start the website in a couple of days. And then in foundation biology we have almost 38 videos so far, and we'll upload more videos on that channel. And we have 118, that is 118 videos in medical lecture. See, you can see 118 videos in our medical lecture, है ना, 118 videos here, and we have 38 videos here. So 118 videos we have in the medical lecture channel. So in this we have immunology, we have human health and disease, ninth standard, some basics of ninth standard also, where we fall in chapter that you can see here. Okay, if we have immunology, we have reproduction, and we have so many videos. We have biochemistry, human health and disease, we have reproduction, then biotechnology videos we have, and we have all human physiologies, mostly like excretory system, respiratory system, and all the videos. We have, good collection of videos. We had earlier more than 500 videos, but because of so many reasons, some videos got corrupted, and we lost so many videos, like more than 350 380 videos we lost. So that much video problem we had recently. So we will shoot again, and we will post, and we will upload the medical lectures and foundation biology videos also in our channel. So please subscribe to our channel, and if you want to know more that what we are doing, we have Telegram channel, and please join the Telegram channel, Learning Life Online. And the website what we have is www.learninglifeonline.com, learninglifeonline.com. So this too, and we have Instagram also. But Instagram, we are not so much into Instagram, because Instagram there are so many things which is like nonsense activities are going on. So we don't want you people to focus on Instagram, because Instagram is not something which we can focus on. So as a student, you can be on Telegram, but recent news said that Telegram, they are going to ban Telegram in India. But I don't know, but still, if not Telegram, we will have WhatsApp also. Take a WhatsApp, we will start once this, if Telegram is not in action in India. So we'll have WhatsApp channel, WhatsApp groups, and we are thinking about WhatsApp channel, that is like Telegram channel, WhatsApp channel also. We'll let you know if anything happens in that way. All right, so that's all. So please join our Telegram channel, join our subscribe our website and YouTube channel. And then let's get started. And we have test series, as already I told you in my previous lectures. We have test series, and that test series, like it is like chapter test, part test, full test. And now chapter test 1 is over, so that's what we are going to discuss. Chapter test 1 questions. Take care. Now I will show you all this data what we have later in future videos, not now. We will start discussing the botany questions. All right, we will start discussing the botany questions, but before that, because students, they were writing, so many students, they continued writing till 6:30. So I cannot take questions, I cannot do analysis. So I will just share the screen of the same question paper, so that we can discuss. So here is the question. This is botany question paper, so we will have botany first and then we'll go with zoology. All right, chalo. So question number one, nobody made any mistake in question number one, or question number two, or question number three. Question number four was also like very easy question, so nobody made mistake in this question. But we have question number five, and question number five also nobody made any mistake. Question number six, and question number seven, question number eight we have here, question number nine, question number 10. Here, few students made mistake in question number 10. So what is question number 10? Let's talk about this question number 10. Meiosis occurs during. So this is one of the very easy question, but I don't know why people made mistake in this question. That is, meiosis occurs during what? So meiosis occurs during gametogenesis is a very very simple question, because in gametogenesis we have two steps. In gametogenesis we have two basic steps. That is, number one is your mitosis and number two is your meiosis, right? So mitosis and meiosis, and this mitosis occurs for the multiplication of the cell, and meiosis occurs for the division of the chromosomes. So during gametogenesis, so that gametes can be produced from diploid to haploid. So this gametogenesis is process in which meiosis occurs. So that will be the correct answer, gametogenesis. I don't know why people, some made mistake in, some marked crossing over, some said body growth, some said tissue repair. Tissue repair I think nobody said, but yeah, body growth and crossing over people said. But the correct answer is gametogenesis. So please make a note that during gametogenesis only meiosis occurs. And because gametogenesis means what? Production of haploid cells from the diploid one, and meiosis is a reduction division. So that will be the answer here. Now, next question, you made mistake here in which question? Yes, question number of chromatids at metaphase. No, this one nobody made mistake. Here, recombination is completed by the end of. So recombination means it's like, please think that we have first is your G0 phase, that is your resting phase, no division occurs there. G1 phase is there, preparation. S phase is there for the chromosomal doubling and formation of sister chromatids and other histone proteins development. So next G2 phase we have for the other protein development for the which is necessary for the next coming mitotic phase. So next is your mitotic phase, where your mitosis can be divided into multiple stages, that is prophase, metaphase, anaphase and telophase. In that prophase we have multiple subdivisions, right? Multiple subdivisions like pachytene, zygote, diplotin, all these 5 steps what we have written. In the previous question you got previous question here, that what is the correct sequence, correct sequence of prophase 1. So that based on that we have to answer this question. Recombination, where recombination occurs? So recombination occurs obviously in the prophase stage. Initial stage, what is the first step of prophase? That is the loosening of the the condensed chromatin fibers. It will get it will initially what will unwinding and winding, so at the end of the prophase 1 all the condensation of this chromatin fibers takes place. Chromatin fiber get condensed at the end of prophase 1. Then in metaphase, what will happen? In metaphase, they will arrange in the they will arrange in a equatorial plate. But before arranging in the equatorial plate, they will get condensed. During condensation, what will happen? They will have synapses formation. Synaptonemal complex will form, and they will have a special protein in between, and they will form synapse. So this synapse formation or synaptonemal complex, that is the stage where your small portion of your chromosomes do crossing over. So that is the stage, and in that 5 stage your which stage is responsible for that? That was the question. Okay, fine. So then you have polysomes, and this polysome several lysosomes. Nobody made mistake here. So botany, most of them they did they made very few mistakes, and most of them they made mistake in zoology. So we'll discuss zoology, what is there in the zoology and what are the mistakes you people did. So rough endoplasmic reticulum, this one only one student made mistake out of this many. So still we have to take this question and we have to discuss. So rough endoplasmic reticulum is ER which bears lysosomes on their surface. Wrong, because endoplasmic reticulum there are two types. One is rough endoplasmic reticulum and another one is a smooth endoplasmic reticulum. And this rough endoplasmic reticulum, they will have their rough surface. For example, let's say we have face, you know, on that face if we get pimples, it will become rough in this region. I have beard and mustache, so it always rough, but still in the region where there is no beard or mustache, if any pimple comes on the surface, if pimple appears, then what will happen, it will become rough. So this rough surface, okay, this rough region or rough surface, okay, this rough surface is just because of the pimple. Similarly, the rough endoplasmic reticulum, they have ribosomes, है ना? So the third option you can see, endoplasmic reticulum which bears ribosomes on the surface, and because ribosome is there, so they will be involved in protein synthesis, है ना? So this will be the correct answer. Alright, so this will be the correct answer, endoplasmic reticulum which bears ribosomes on the surface and is involved in protein synthesis. That will be the correct answer. Alright, chalo. The next question. No, this one nobody made mistake. People are very smart. You can understand that this is Golgi apparatus, right? Cis phase, trans phase, Golgi apparatus. Alright. So Golgi body, what Golgi body do? Golgi body remains in close association with endoplasmic reticulum. Yeah, that is true. And materials to be packaged as vesicles from the ER which fuse from cis phase and move towards the trans phase of Golgi apparatus. Cis and trans phase, that you have to think, है ना? Cis and trans phase. What is cis and trans phase? Nobody made mistake, but still I thought, okay, this we can explain. Cis phase and trans phase, the 1A region which is given in your NCRT also, okay, which enters and one through which leaves. Take, so whether this is correct or not. First one is absolutely correct, but second one is correct or not. Just think, go through that NCRT, that exact line is given. So that will be the answer. Take it, the membrane bound vesicular structure, nobody made mistake. This one also nobody made mistake. So you people are very smart. Powerhouse of the cell, tonoplast. This one tonoplast, it confused few people. Like most of them, they took time in this section, and so that because we get analysis and in that we can identify whether you took time or not. So here in this region you took time. So some students, they took time in this region just because maybe because of thylakoids and tonoplast. And even because of this powerhouse of the cell is there, suicidal bag is there, lysosome. So maybe that created confusion, but there is no confusion. Powerhouse of the cell means mitochondria. One is C. If one is C, only one option is there. Okay, which one? Second one, second option. This one is the only option we have, है ना? This one is the only option we have. So this is second one. Now, but still, what is tonoplast? Tonoplast is present in the vacuole, that is a membrane, of vacuole, that is a tonoplast. Thylakoid, chloroplast, suicidal bag, lysosome. So very simple question. I don't know why people made mistake here, but yeah, very simple question. Chalo, the next question. Here, amyloplast means what? Elaioplast, eleuroplast, and chromoplast. Chromoplast, obviously pigment. So 4 is C, then this will be the correct answer, right? Okay. Yeah, sorry. I'm really very sorry. I got some issue with my camera. Yeah, and I hope that your screen is visible. We'll discuss zoology. I don't know what happened again. Still, it's blurry. Okay, but that's okay. Next, I'll try to sort out all these issues. What is coming today, because this is the first live, and technically, I'm not very sound. I'm good in subject, but technically I'm not so sound. I'm really very sorry for this technical glitch. We'll continue. We'll continue with the zoology, because that's all. That's the mistake you people did in botany. So next we will go with zoology. So here in this zoology portion, we can say the questions which you made mistake will be, or will slowly shortly we'll discuss all the questions, and the question you which made mistake, that one alone will discuss in detail. So the first question, which are the following is the applications of biotechnology? Biotechnology application means application in medicine is there, application bioremediation is there, then pharmaceutical, therapeutics and diagnostics. All these we can have. Genetically modified crops and processed food also we can produce. Waste treatment, energy productions, all these we can use biotechnology in this. So we can write accordingly. So this question nobody made mistake. The critical research area of biotechnology, pure NCRT line. So critical area of research, what is that? Downstream processing technology to purify protein and organic compound, creating optimal temperature and optimal conditions through engineering from the catalyst to act, providing the best catalyst in the form of improved organisms, usually a microbe or pure enzyme. So what is the critical area? Critical area of biotechnology means where we need to work very consciously, which it will not be helpful if we are not focusing there much, and we have to develop more. That is, the development in that area is required. So NCRT line, no need to discuss much in this question. The plants, bacteria, fungi and animals whose genes are altered by manipulation are called, yes, genetically modified organism. That is the correct answer here. Transgenic organism, transgenic organism is also correct, but transgenic organism means we can say where we are manipulating the DNA, alteration. So alteration by manipulation are called, here also it's given. So transgenic animal is also correct. So both A and B should be the correct answer here. Genetically modified organisms or transgenic organisms. Now, some strains of Bacillus thuringiensis have proteins that can kill what? So here Bacillus thuringiensis BT cotton. BT cotton means that what? Plant feeding that beetle. So plant feeding beetle. So tobacco bollworm is there, budworm, bollworm, tobacco plant also we have crygenes, beetles, crygenes. So that you have to think, what is that? Cry protein. So cry protein which will cause damage in the cotton plants, in the tobacco we have a nematode, so we should not go with that. So beetles, coleopterans, dipterans, flies and mosquitoes, no. Tobacco, budworm, armyworm, we have beetles, so because which eat that leaf, and that will be killed here. So what is true about Bt toxin? Bt toxin, what is true? The concerned bacillus is antitoxin. So this question nobody made mistake, so we should not waste time on that. Proteins encoded by the genes Cry1AC and Cry2AB control what? So cotton bollworms and corn borer, budworms and butterflies, NCRT line, 1AC and 2AB. This is for only one thing, and we have 1AB, 2AB, 2AB, and we have 1AB also. So that is cotton bollworm and corn borer. So we have to choose which one is the correct answer. NCRT, just above the RNA interference topic, it is given clearly. Go through that. So see, I can help you. I'm just helping you in order to reach the correct answer. But NCRT you have to go through 100% for biotechnology, cell chapter, all those chapters. A nematode that infects the root of tobacco plants. Yes, nematode. What is the name of that nematode? It's Meloidy gyne incognitia, and this is a nematode. And in the principle or the process, what we use here is RNA interference. the next topic what we have here. Which of the following genes do not occur naturally in living organisms? So that will be, nobody made mistake here. So we can leave that question, but still this is also an NCERT line, pure NCERT. Just go through that. And a dicotyledonous plant forms crown gall when Agrobacterium tumefaciens come in contact, or Agrobacterium rhizosome, specific part of Ti plasmid, or specific part of DNA of Ri plasmid gets integrated with the plant chromosome. So a dicotyledonous plant. See, we have different vectors we use, right? We use different vectors for that. Agrobacterium tumefaciens is something like we can use it as a, what we can say, Agrobacterium tumefaciens is a kind of vector we can use, है ना? The transformant we can use. Fine. So that will be the answer here. So the strategy used to prevent the nematode infection in the root is RNA interference. That's the answer of this question. Because RNA interference only we interfere the mRNA and we block the development or the formation of the protein, which can cause damages. So the first human hormone produced by recombinant DNA is your insulin. That is NCERT line, pure NCERT line. It was given, and I think nowadays, in the latest NCERT, I'm not sure whether it is present or not, but the correct answer will be your insulin only, the first recombinant DNA technology produced hormone. So that is your insulin. Pick out the incorrect statement. Insulin consists of two polypeptide chains that are linked by hydrogen bonds. No, that is disulfide bridge. So disulfide bridge will be the correct answer here, not hydrogen bond. Insulin is used to manage adult onset diabetes. Adult onset diabetes means type 2 diabetes they are talking about, which means the diabetes which occurs after 18 years of age, that is type 2 diabetes. So insulin is used to manage those, that depends like insulin deficiency is there or not. If insulin deficiency is there, so insulin can be used for that. It is possible to produce human insulin using bacteria. Yes, it is possible to produce human insulin using bacteria, means as a vector, not the gene of gene what we have to use. As a vector we can use and we can produce. In mammals, insulin is synthesized as a pro-hormone, that is pro-insulin. Yeah, that is correct. Pro-hormone in the sense inactive insulin will be produced as, and they will be like after the production, whenever it's necessary, it will be broken down and it will be so we mature. So it gets mature by removal of one peptide strand of insulin. So which one is the correct incorrect statement? That is the question. So first option is the incorrect option because it is not hydrogen bond, it is disulfide bond. Alright, so the next question will be, so this question you made, so many people made mistake in this question. So the correct answer should be the A answer because of the hydrogen bond. Okay. The pro-insulin contains an extra stretch. Nobody made mistake in this question, so nothing to worry about. The first American company, Eli Lilly, so no issue at all. First American company, NCERT line, Eli Lilly will be the correct answer. The first clinical gene therapy was given to a 4-year-old girl with which one? That is your adenosine deaminase deficiency, NCERT line, which means bubble boy syndrome. We have studied all those things. So adenosine deaminase deficiency, like lymphocyte treatment will be done. So 4-year-old girl, NCERT line. So ADA deficiency will be the correct answer. Next, 16th question. The genetic defect adenosine deaminase deficiency may be cured permanently by. So permanent cure, it is not possible once it is like once the baby is out of the uterus. So we have to administer adenosine deaminase through injection. No, that is not the permanent cure. Bone marrow transplantation can be the permanent cure, but all right. But still, bone marrow transplantation is not 100% we can say permanently cure according to NCERT. Introducing gene from marrow cells producing ADA into cells at early embryonic stage is the permanent cure. Take it, because enzyme replacement therapy, bone marrow transplantation, how long you will do? And after the birth, it's a very risky procedure also, bone marrow transplantation. And I don't think that will be a permanent cure. And according to NCERT, D is the correct answer. So what I think doesn't matter, what is given in NCERT, that is important. Right? So that's the thing. Next is your PCR. PCR can be used for a detection to detect HIV in suspected AIDS patient, to detect gene mutations in cancer patients, to identify many genetic disorders. PCR is just for amplification, है ना? PCR is for amplification, but we can use for cancer also, we can use for genetic disorders also, we can use for HIV also. So all of the above will be the correct answer here. ठीक है, in NCERT it is given for HIV and cancer, and many genetic disorder we can use PCR. Why? Because PCR means polymerase chain reaction, and polymerase chain reaction will be used to amplify the DNA. And if there is any mutation, by adding a probe we can identify whether they have mutation or not, they have genetic disorders or not. ठीक है, so mutational genetic disorders we can identify. So all the above should be the correct answer here. But what I suggest you to refer your NCERT, what is given in NCERT in molecular diagnosis topic, it is given in the third paragraph or third paragraph or fourth paragraph maybe. To detect HIV is given, obviously it is given in cancer also. We can use this PCR for detection of cancer. So more than one option is correct. So we will confirm that all the above will be the correct answer. So sometimes hard work will not work, so smart work we have to show. So smart work will save your time also, and it can give you, most of the time it can give you the correct answer. Sometimes when your luck is not too good, it can give you the wrong answer, but still. Transgenic animals are the animals whose genome has been altered by. Transgenic animals are the animals whose genomes has been altered by deletion of one or more genes from them, introduction of a foreign gene. NCERT, in NCERT, it is given that addition or deletion, introduction of a foreign gene by manipulation, we can say. Introduction of a foreign gene by manipulation, that should be the correct answer. Because additionally we add our gene of interest, and that will, that will start working. So transcription translation as a normal gene. Okay, so deletion, we don't generally do deletion or addition and deletion by gene manipulation. Introduction of a foreign RNA we don't do. Deletion of one or more gene from them we don't do. But introduction of a foreign gene by manipulation, that is should be the possible correct answer. Okay, and one student made mistake in this 18th question also. Transgenic animals are extensively used for all of the following procedures except chemical safety, vaccine safety, toxicity. Nobody made mistake in this question. It's a very easy question, and this one also very easy question. Nobody made any mistake. First transgenic cow was Rosie. Nobody made mistake here. This one also nobody made mistake. Wrong statement, they have to select. 23rd question, which organization was set up by the Indian government to make decisions? Very simple question, nobody made mistake in this question also. So we'll go with 24th question, nobody made mistake in basmati rice. This one, all these questions are correct, and yeah, here directly we can move to 37th question, which you made mistake here. When dash is added, purified DNA precipitates outside the collection. It's a very simple question, but some wrote glycerol. Come on, we don't use glycerol for precipitation in DNA. Glycerol, and one student wrote chilled NaOH. Chilled is fine, but not NaOH, sodium hydroxide. No, we are adding ethanol. It's NCERT line, and you did practical also. Yeah, come on, 12th standard. Take care. So very few made mistake in 37th question, and then 42. Next question which you made mistake is 42. Here, yes, this type of question, it can create confusion. Amplification of gene of interest by using DNA polymerase may go up to 1 billion copies. That is NCERT. In the just diagram, below the diagram it is given, and in the, I think the theory part is also given. So in the PCR, it is given how many copies are produced at the end. Alright, billions of copies are produced. So here 1 billion copies are produced by amplification. So PCR can produce that much copies, not million, billion. Next is 44th question. You made a mistake. 44th question, sequence. So sequence, I don't know why you made, only one student made mistake. I think just in a hurry you did, and that's why you made mistake. So the first step will be denaturation, second step will be annealing, and third step will be extension. So first it will break, then primer will attach, and then you will get extend. That's all. 2 copies will be produced here. Alright, and then 45th question, in PCR the process annealing means. Annealing means what? Addition of the primers, we can say. The joining of 2 primers at 52 degrees Celsius at the 3' end of the DNA template. In the template we are adding 3', which means we are moving towards 5' to 3'. So 3' on the template strand we are adding, not on the 5' end. Okay? Generally DNA moves in the 5' to 3' direction. So on the template means opposite side we have to add, है ना? So opposite side means what? 3 prime we have to add, then only it will have 5 prime starting, and then it will move towards the 3 prime end, right? So this will be the correct answer, second one. All right, so yeah, that's all about the zoology. Also botany, zoology, we are done with the botany, zoology part. And I think we should talk more about this you know classes what we have, you know, here. So this channel, join our YouTube channel, join our Telegram channel, and uh, in the Telegram channel we have courses. So join our courses also, so that you can get benefited. ठीक है, join our courses, join our test series. So first chapter test is over, and second chapter test, I learned one lesson, very important lesson. What are the different technical problems we can face during this online sessions? Our team is working, and we will definitely have a better, better live streaming next time. All right, so please join our Telegram channel, and our just keep an eye on the website also, because anytime we can launch, and anytime it can come live, so you can access all our videos, exclusive videos in that website. All right, so thank you so much for joining, and I hope you will visit our channels, that is Foundation Biology, NEET Biology channels, and all the videos of those and of these channels. All these like I did shooting of this all these videos in back in 2020, so only whiteboard and marker, that kind of system we have there. Sometime I think very few videos we have digitally I took, and but nowadays I'm going directly, like what we can say, digital. We have all the setups here, so all clarity and no issues will be there in the sound clarity. You can see the live, sound clarity will not be the issue, and the you know clarity of writing also, so the words what we have. So do share this information with your friends, friends, and like this video if you really liked this session. And I'm really very sorry, I apologize for all the technical difficulties what we faced today, right from the beginning, right from the 6 o'clock, and in between also we had so many problems. And I'll rectify, and definitely I'll make sure that next time we'll give the best of the best. All right, and if you have any problem with this channel or joining, or if you have any feedback also, you can directly contact me. This is my number. You can directly contact, and you can share your feedback, you can share your valuable feedback. And if you want any, if you have any query, you can ask. This is you can text me on WhatsApp also, you can text me on Instagram, Telegram, wherever you want, and feel free to ask any question. All right, and if you have any special request also regarding the channel, regarding the videos, if you want any lectures, a special request on lectures, you want to if you want me to shoot and deliver to you, we can also do that. And yes, we have one more product, and that we are not promoting that product, and that product is like question bank. We are having question bank, and we are selling it on a very affordable price, so you can purchase that also. But still we are not promoting that, because the work is going on. Only biology is ready now, physics somewhat half of the physics is ready, chemistry is still working, we are working on that, so the product is not completely ready. But as soon as it it comes in the market, will definitely I'll notify you. So keep keep going through the Learning Life channel and the Telegram channel for the latest updates and all the products. Okay, thank you so much for joining, and have a great evening ahead. Thank you so much. Bye bye.